Hello A-level student, my name is Dukunimana Paul. So after realizing that some students are getting higher to get good grade in biology, which is A positive, so I decided to make a video from senior 4 up to senior 6. What I'm requesting you is to do subscribe on this channel so that you are going to get all video I'm going to put on this YouTube channel, and then at the end the result is going to get a good marks or good grade in the biology. So in the previous video we have talked unit number 1. In this video we are going to talk about unit 2, which is called introduction to classification. So the students who are doing MCB, who are doing PCB, you should come and sit with me in this video, which is not going to be too long. So I'm requesting you to come here, then take your pen and even your notebook so that you can continue with making biology fun. So the above content of today, it's all about the introduction to classification. I'm not going to delay by explaining this type of content, but I'm sure that each things which is shown here, I'm going to explain very well in the detail in the following the slides. So the unit called introduction to classification. So when we say classification, just you are understanding the word called class. Means you're going to make organism, living organism into their classes. Means some organism, you know, are called plant, other are mammal, others mammalia, other protista, monera. Means those things you already know, but those are the classes. Means the study of the classification means it is the way you are going to make your living organism into specific classes according to their physical characteristics, morphology, and so on. So the care unit competence of today means after ending this video, a student is able to apply basic knowledge of the classification to group living organism into 3 domains. So you will understand what domain it is. Learning objectives also are quite many, but I'm going to be quick so that we can make them easier to us. But at the end of this video, learner is going to be able to understand what is kingdom, what is domain, kingdom, phylum, class, order, family, and genus. So, and also the outline, the features of the 3 domains: archaea, bacteria, eukarya. So a student will be able to draw and rubber the structure of the bacteria. Student also will be able to identify common bacterial diseases in plants and animals, and also to outline the features of the kingdoms: protista, fungi, plantae, animalia. And also the student will be able to explain why viruses are not included in the 3 domains classification. Also classify the virus based on the nucleic acid type. Also to demonstrate the role of the bacteria in dairy production. Discuss the method to prevent the common bacterial diseases. And also to develop observation skills and design dichotomous. So this is the virus, structure of the virus, so you will be able to explain. And also this one, it is the sample of one diagram which I will use to make the dichotomous key very easy, like piece of cake. So thank you. This is like our learning objectives. So it means that learner is going to be able to explain all of this. So be with me. I'm going to be quick, but not too quick. So I make things very well to be understandable by every student who are understanding me. So now, number 1 things you have to look at in this unity, it's all about taxonomic hierarchical. You know, you have seen this word many times. What you call hierarchical classifications means they're asking you what is hierarchical classification. So before explaining this, you should understand what is taxonomy. When you say taxonomy, taxonomy is a science of naming and classifying the organisms. Means taxonomy, it is the science of naming and classifying organism. Means in classification, the science which study classification is called taxonomy. Means taxonomy is the science of naming and classifying organism. Means to say that this is animals, this is a plant, this is what means that is what you call taxonomy. Means taxonomy means once you are taxonomist, you are people who have studied to name and to classify organism. So thank you. Now, when we say this science of naming and classification organism, first thing to understand is the taxonomic hierarchical levels. Means in the classifying our organisms, we should be based on some levels to make sure that all organisms are included in our classification. So here there is introductory activities. For instance, there is a continent, district, country. Here they just said province, sector, village, and family. So means my task is to ask you, can you arrange this by sizes? Thank you. So in this size, you're going to arrange from the highest to least population. Means which between the continent and district, which one is having the highest population. So by the answer, as you know, the continent, like we have Africa, means Africa, it is big. Now after continent, usually we have country. After country, remember, we have the province. After province, you have district. After district, we have a sector. After sector, we have cell. Now after cell, we have a village. After village, now we have a family. So based on this arrangement, we are coming from large to the smallest. The continent, there are many people there, many countries, like even the culture are different. As you're going down to your family, you are being specific. So in the same way of arranging this one is the same way we do what you call taxonomic hierarchical classification. So hierarchical levels are domains, as you have continent, kingdom, as you have country, phylum, as you have province, class, order, family, genus, and species. So by using these levels, we can know which animals we are talking about. Means after putting the classifying organism based on those levels, we are able to say this is Homo sapiens, not pig. So like for example, when you put also in this introductory activities, let me suppose that we are talking about the people called XICXI. So these people usually is Rwandan from any district or any cell. So when we go to the level of the continent, you cannot distinguish Paul, who is called XIC, sorry. You cannot distinguish these people called XIC from maybe Sudanese citizen. No, at the continent level, they are the same. When you come to the country level, now the Paul, which is XICXI, is different from Sudanese people. So, but you are not able to distinguish this people called XIC from another. Maybe in the country, there is many provinces. It means as you're going deep until to the family of this patient, for these people, you are able now to describe this organism or these people. So I hope you are understanding regarding to those levels. So you should know those levels as the way to arrange. If they ask you to arrange them, you should arrange accordingly. Take these things. It is very, very, very important. If they ask you to arrange from the largest to the smallest, try to write domain, kingdom, phylum, class, order, family, genus, and species. Very good. So key point to keep, as you move down hierarchy, the number of individuals decrease. Okay, means on this domain or kingdom, let me say kingdom animalia. Many animals, many organisms are there. But when we reach here to the species, when we say like Homo sapiens, only few individuals are there. So, but shared features increase. Means as you are decreasing down, number of organisms are decreasing, but organisms you are remained, they have similar characteristics. Like human being and gorilla, they separated maybe at the family level, where gorilla they have its own family, and then human being also have the family. So at this level, as you are down the group, as you are making down here, the organism, number of organisms are decreasing, but the characteristics between the organism are increasing. So I hope you are understanding this taxonomic hierarchy. Very, very important at your national examination level, either multiple choice or wrong question, or to arrange. Thank you. So other things to say, it's about the binomial nomenclature and the taxonomic hierarchy. So to mean that each organism, they should have name. As you who are understanding this video, also you have name. So binomial nomenclature is the scientific naming system is induced by Carl Linnaeus. So this is a scientist who introduced the system to name our living organism. So this system introduced by Carl Linnaeus of binomial nomenclature is the aim to give the organism two Latin names. Maybe it's why people also they have two names, but in this generation people they are adapting to have more than two names. So according to this binomial nomenclature introduced by Carl Linnaeus, he introduced the system which said that organism should have two Greek or Latin names. There's two names. There should be genus and species. Means genus name and species name of the organism, they should be their names. How are arranged? So the organism should be started by genus name followed by species name. So usually this genus name should be capitalized. It should start by capital letter followed by the species name which is lowered case. Means if say like sapiens, this one, if it is species, should start by small letter. Now to make this binomial nomenclature means like you have to say Homo sapiens. Means what this Homo means, this Homo is species. Now what this this Homo, it is genus. Now this sapiens is species. When we come here on this classification of the earthworm, when we say Lumbricoias terrestris, means this one is the genus name. Now this is species name. So the binomial nomenclature, it says that genus name starts with capital letter. Now species name starts with small letter. When we come here to hibiscus, it is Hibiscus rosa sinensis. Now this is the scientific name of hibiscus. So this hibiscus, this is the genus name, is started by capital letter. Now this one, it is a species name which is started by small letter. So I hope you are understanding what you call binomial nomenclature. So binomial nomenclature should keep in head that the names of organisms should be Latin names. Those Latin names, one should be genus followed by species. Genus should be started by capital letter. Now species should be lowered letter. So I hope you are understanding. So you should ask yourself, why do we need to name why binomial nomenclature are important? Why to name the organism are important? So it is important because we should precise identification. If we say we need this organism, for example, human being, it should have his or her own specific name, so that you can distinguish these individuals from other organisms. So other things, it is international recognition. So those names are not Rwandan names or Ugandan names or any culture names, are Latin names. Means each organism you are seeing, they have international name, which is in form of Latin names. Not like you can say pig. Pig is not a name. Pig should have specific name. You should search on your internet. So the benefit of the benefit of this binomial nomenclature is precise identification. This is number one. Means you should identify organism accurately, and also international recognition. Means once you are saying Homo sapien, either Ugandan can understand that it is human being, either Egypt, they can understand that it is human being, even India, even China. Means international recognition. Means The importance of this binomial nomenclature is precise identification and international recognition. Thank you. At this level, after understanding this, we can start this taxonomic hierarchical classification levels. We have seen that the level number one, as we have seen, level number one is domain. So you should understand what are domains. What are domains? Okay. What are kingdoms? What are phylums? What are classes until to the species? So by starting two domains, we have only three domains in this world. Only three domains are there. Eukarya, bacteria, and we have eukarya, bacteria, and eukarya, bacteria, and archaea. So those are the three domains of in the taxonomic hierarchical classification. So three domains of the organism. Number one is bacteria. I hope you know bacteria. Usually it is a prokaryote. Prokaryote. When we say prokaryote, we mean there is no nucleus, no true nucleus. Means there is no nuclear bond membrane, and also their DNA are circular. You should memorize that prokaryote, no nucleus, and circular DNA. When we say prokaryote, usually there is no nucleus. Now to mean that their DNA are circular. Where the difference between bacteria and eukarya, see, this one bacteria DNA is circular, but here in eukarya the DNA it is linear. You see this difference? You should keep it and memorize, because this is where multiple choice questions are coming. So you should memorize this. So and also bacteria, apart from prokaryote, apart from that they are prokaryote, no nucleus, they have also circular DNA, but also they have cell wall. Their cell wall are made of peptidoglycans. Very, very important to keep in the head. So, bacteria, I hope you are understanding. Number one, bacteria are prokaryote. Number two, bacteria, no nucleus. Bacteria, they have circular DNA. Bacteria also, they have cell wall made of peptidoglycans. So, once you know bacteria, eukarya is going to be very easy for you. Very, very easy, because all of them, they are prokaryote. All of them, they don't have true nucleus. All of them, the DNA are circular. But when you come here on the cell wall of the archaea are not made of peptidoglycans. I hope you are seeing the difference. Okay? I hope, and also this archaea, there are also other specific difference from the bacteria. Those archaea organisms are extreme condition. Means they can live in hot spring. They can live in area with high temperature, area which is acidic, without dying. So I hope you are understanding the difference between bacteria and the archaea. All of them, they are similar except two. This archaea, they don't have peptidoglycan cell wall. This archaea, they can survive in extreme environment, like environment with high temperature, environment which is acidic condition, environment which is salt environment. So I hope you are understanding the two. So this one, eukarya, usually it's like eukaryote. It means they are eukaryotic cells. They have eukaryotic cells. Means, you know, eukaryotic cells, they have the true nucleus. They have many organelles which is bonded by cell membrane. They have linear DNA. The key difference, linear DNA. Here there is circular DNA. Here there is no organelle bonded membrane, but here in eukaryote we are having the membrane bond organelle. Means the organelles, nucleus, mitochondria, chloroplast are bonded by membrane. So I hope you are understanding this one very clearly. Let me give you an assignment. Here there is what are three domains of living things. I hope no one who can fail this question. I hope no one. So there are three, eukarya and so on. So discuss the way in which the domain differs from the kingdom. I hope you know from that way, you know, the levels are like this one. When number one is the kingdom, sorry, domain. Number two is the kingdom. Means one difference means domain, they have many organisms there, but kingdom, the few organisms are there. And also when you come back to the way of the sharing the characteristics, in the domain, the organism there are not usually they are not similar with characteristics. We have seen as you go down, characteristics similarity increase, but the number of organism decrease. I hope you are understanding. So all of these are the question. I have here the answers. You should pause and review and then understand very well so that you're going to get a positive grade in biology. Thank you so much. I hope you are seeing those are the question, those are the answers. See, pause and review. Pause and read question number 1 to up to 5 and then see the answers, and then so that you are going to have a positive grade in biology. Thank you so much. So now it's time, after finishing the domain, let me talk about 5 kingdoms. 5 very, very important. So, and even in my national examination, they have asked me the question like this, asking me what are the difference between kingdom Plantae, Fungi, what are the difference between blah blah blah, and also they can give you many multiple choice question. Among the following kingdom, which one have chloroplast? Plantae. Which one they have the cell wall made of cellulose? Usually are Plantae. Which one they have cell wall made of chitin? Is Fungi. Which one which have cell wall made of peptidoglycans, bacteria, and so on and so forth? What are the examples? I hope this diagram it is understandable and very clear, very clear. You should pause and review and understand it very well. So let me have a look on those five kingdoms: Monera, Protista, Fungi, Plantae, and Mammalia. So usually, first of all, I'm requesting you to take examples. But before you have to take examples at least. When we say Monera, bacteria. Monera, bacteria. Monera, bacteria. I hope you are understanding. When we say Protista, there are many examples. Usually Protozoa, Arc, Paramecium, and so on. So fungi, there is Mushroom, Rhizopus, there is a Muco, Aspergillus. So many examples of fungi are there, but here I put you mushroom, you should take. Plantae also, they are, you can say beans, you can say banana, and so on. Means plant are quite many. And also Animalia, examples are mammals, and even you are Animalia, so don't complicate life. So at this level, before talking about any characteristics of this, after means like Monera, bacteria, Protista, protozoa, Fungi, mushroom, Plantae, banana, Animalia, you. Thank you so much. So those are the examples. First of all, take examples. After taking examples, you should come here and then understand this chart I put here. It is very, very important. I want to highlight the things here. Very, very, very important. Once you say there is plant, Animalia, Fungi as kingdom. All of this, they have 2 characteristics which are common. All of them, they are common. Once we are talking about multicellular, this one, and this, all of them, they are multicellular. I hope no one who will tell me, sir, I don't know the characteristics of Plantae, Animalia, or Fungi, because number 1, all of the 3, they are multicellular organisms. I hope it is clear now. So now, when you come also for the next, I know that the cells, they can be either eukaryotic cells or prokaryotic cells. All 3, they are eukaryotic cells plus, these guys, they are, yes, Protista, it is here, means all of them, this one, but this Protista don't take, because sometimes this Protista, they are multicellular, other they can be unicellular. So means we talk about just only the Plantae, Animalia, and Fungi. All of them, they are multicellular and they are eukaryote. So remember, when we say bacteria or we say Archaea, all of them, they are unicellular and they are prokaryote. Usually this one they take as bacteria, but this one, as you remember, Archaea, they can survive in extreme environment, and then remember they don't have peptidoglycans in their cell membrane. So, but when you come here on the bacteria, they are unicellular and also they are prokaryote. So, means all of them, they are the same as I told you. They are different, they come on their cell wall and the environment to survive. But or they have the circular DNA, okay? All of them, they don't have nucleus, things like that. Means Plantae, Animalia, Fungi, all of them, they are multicellular. And then all of them, I should keep this, means all of the three, they are multicellular and they are eukaryote. While all of this one, bacteria or archaea, they are unicellular, and the same case they are prokaryotic cells. Means when we say multicellular, means organism is made of more than one cells. More means many cells, multi, more, many cells. But when you say unicellular, only one cells are there. Only one cells are there. When you come to the level of protista, they are in between. Usually they are eukaryote. But when you reach to the level of multicellular and unicellular, they can be both. Means we have protista which are unicellular, for example, yeast, and protista. I'm sorry, yeast, it is fungi, it is not protista. Like a unicellular, they are like amoeba. Amoeba are a unicellular, which is protista. And we can have multicellular like a helminth. Helminth, you can say hookworms, things like that. No. But there are some protista which are multicellular. You should search examples. So I hope now those are the characteristics which are common. And then once they give it to differentiate, can you differentiate fungi from bacteria? Yes, I can. Fungi, they are multicellular, they are eukaryotic. But while the bacteria, they are unicellular and protista. But now the problem, when they ask you to distinguish between plant and fungi, what will you do? So I need, you have to continue to sit down. It means the characteristics is not ended by here. So I hope now when you come here, you are understanding that when you say plant, animal and fungi, all of them, they are eukaryotic, all of them, they are multicellular. While the Monera, they are prokaryote and they are unicellular. When you come to the protista, they are there between. They can either be multicellular or unicellular, but they are usually eukaryote. I hope you are understanding everything here. Now let's go to the kingdom one by one. I hope you are enjoying. If you are enjoying, make subscribe, comment and share. Means you can tell me what I can change, and then I hope you are understanding this very well. So thank you so much. So let me start kingdom number one, which is Monera. Usually I told you when I said Monera, you should understand bacteria. So we have already seen that in the Monera that they are unicellular, and that Monera they are unicellular and they are prokaryotic cell. So I hope the two characteristics are there. What I'm emphasizing you to know, it's about the main parts of the bacteria. So let me have color. So bacteria, usually bacteria, they have two envelopes. Means envelope number one are cell wall. Okay, this is cell wall. Envelope number two are cell membrane. I hope you are understanding. Okay. So inside the bacteria, there are genetic material, can be either DNA, and then remember is circular. I hope you are seeing here. This can be either DNA. Yes, they are always DNA. Okay, which is circular. Remember, and also there are some ribosome. In the bacteria, there are small ribosome. In this bacteria cytoplasma, they are ribosome. Okay. Now, always there's part I'm putting here exist in the bacteria. But some bacteria, they have envelope or capsule, other they don't have capsule. Some bacteria, they have what you call flagella, which for motility, other they don't have. I hope you are understanding the part of the bacteria. So take time and pause and review. This part of bacteria can be important. Maybe they can ask you like importance of cell. They can draw, then put A here or B, then asking you to name rabo part A and B and so on. So the characteristics of this kingdom Monera, number one, they are prokaryotic cells. I hope you have understood. Not true nucleus. They are single-celled, to mean that they are unicellular, means the smallest to simplest organisms. So they have diverse shapes, means they can be either sphere, rod, means when they are sphere they are called coccus. When they are rod shaped they are called bacillus or spirilla. Means some, let me also again have the color again. Some bacteria, they have the shape like this. When the shape is like this, we call this one as the it is spherical. Okay, this is spherical. It can be either spherical or it can be either rod shaped, or it can be spirilla like this one. I hope you are understanding. Spirilla means it is curved in this way. So the reproduction of the Monera usually always are asexual reproduction. The cell wall, often they contain peptidoglycans, means bacteria, remember, except those which are archaea, which don't contain these peptidoglycans, but their cell wall, this one, they contain peptidoglycans. I hope you are understanding. Means their metabolic diversity include autotrophs and heterotrophs. Means they either they can make their own food, or they can not make their own food, means they can occur as parasite. So take time and review the characteristics of kingdom Monera. Take time, it is very, very important. First thing to know, I told you always two characteristics, are prokaryote or eukaryote. This one is prokaryote. Are them unicellular or multicellular? As you are seeing here, it is only one cell, means it is unicellular, only single celled. So are them autotrophic or heterotrophic? Means they can be both. They can either make their own food by photosynthesis, or they can search their own food. So they have cell wall made of peptidoglycans. Their reproduction are usually asexual reproduction. I hope you are understanding kingdom Monera, kingdom number 1. I hope we can go to the kingdom number 2. Kingdom number 2 is called kingdom Protista. I have started by usually unicellular organism. You are seeing here, those are examples: Amoeba, Paramecium, Chlamydomonas, or Varvos. All of this, you see, they are only single organism. This one, only single cell. Only single cell. Now this is now organism. So when we say unicellular, this is what we are talking about. It means when they ask you characteristics of the Protista, once said, characteristics of kingdom Protista, they are unicellular, number 1. But you have seen they can be either unicellular, but we have some which is multicellular. So let me talk about the characteristics. They are usually eukaryotic cell, means they contain true nucleus. Mostly they are unicellular, but you have some which are multicellular. Okay, means they are a heterogeneous group, include the protozoa, and then large, means they are autotroph. When we say autotroph, they are plant-like. Autotroph means they do their own, they make the photosynthesis, means they make their own food. Or they can be either heterotroph, to means that they don't make their own food. When we say that algae is example of the Protista, now algae, they have the chlorophyll, green pigment, means they can absorb the sunlight and then they can make the photosynthesis like a plant. That's why this is called plant-like. So they have the cell wall made of cellulose. Other do not, means it is not all of them which have the cell wall. The way of nutrition, okay, as I said, they are autotrophic or they are heterotrophic, means this one it is kind of repetition. This one they are the same. In terms of the reproduction, we have seen that the Monera are usually asexual, but this one they can be either asexual or sexual reproduction. So I hope you are also understanding those characteristics of the kingdom Protista. So by understanding that the kingdom Protista, always two characteristics are there. Either they are eukaryote or prokaryote. This one it is eukaryote. They are unicellular or multicellular. This one they can be both unicellular or multicellular. I hope you are understanding the characteristics of Protista. So when we come to the kingdom number 3, called fungi, you know, this is an example of the mushroom. Those are the kingdom fungi. Usually the way of reproduction are called formation of spores. Those are spores. Means these spores, if it fall here, means they are going to be grow of new fungus. So I hope you are understanding. So by time of describing their characteristics, you know, two always are there. We can say they are this one, they are multicellular, you know, it is not one cell, and also they are eukaryote. I hope you are understanding. So means they are eukaryotic cell, means true nucleus are there and the membrane bound organelles. They are heterotrophic organism, means they don't have chloroplast. They obtain food through decaying matter. Usually fungi, they use what we call saprophytism. Saprophytism means it is feeding on dead matter. So means the kingdom of fungi also they contain cell wall. Their cell wall again and again and again is not made of cellulose. Cellulose or peptidoglycans. Their cell wall is made of chitin. So usually the cell wall of fungi is made of chitin. Take this one, you will see this one either in multiple choice. Usually in nowadays NESA has changed the way of asking. Section A 70 marks are always multiple choice. So keep this one, that cell wall of the fungi are chitin, not cellulose, not peptidoglycans. So their ways of reproduction usually are spore formation, but also they can be what we call hyphae. They can be either hyphae or they can also form what we call budding. So like budding, usually it is formed in yeast, budding yeast. So means when they ask you about the multicellular or unicellular, tell that the kingdom of fungi they are always multicellular, but there is one exception called the yeast. So yeast it is only fungi which is unicellular. This one yeast it is only fungus which is unicellular organism, while others they are a multicellular organism. I hope you got it. Means body structure, they can be either unicellular. If it is unicellular, it is yeast. But other remainings are a mushroom. Means remember that we have said that fungi they are only multicellular. Keep in that they are multicellular organism, but there is only one exception, which is yeast. I hope you are understanding. So and also other characteristics, it means they don't have chloroplast. Fungi means the difference between fungi and the plant, they don't have chloroplast. Means there is no chloroplast, means there is no photosynthesis. If they ask you the difference between fungus and the Monera, you know the difference is easy. Monera they are unicellular, they are unicellular, they are prokaryote, where this one are multicellular and prokaryote. You know, they are unicellular and they are eukaryote. So those are the characteristics of the fungi. They are eukaryotic cell. They do heterotrophic nutrition. They have cell wall made of chitin. Mode of reproduction are spore formation. They are usually multicellular organisms, but one exception which is yeast, unicellular, or they don't have chloroplast. I hope you are understanding the difference between fungi and other kingdoms. Pause and review. So let me come back on the fourth. Let me come back on the fourth kingdom. Let me have the marker. So this is the fourth kingdoms. This fourth kingdom, it is called Kingdom Plantae. So I hope you know kingdom of plant, it is common. In nowadays we are seeing the plants. means they are a eukaryote, as we have said. They are eukaryote and they are multicellular. They are autotrophic nutrition, to mean that they do photosynthesis, means they have chloroplast. They have cell wall, not made of chitin as fungi, not made of peptidoglycans as bacteria. The cell wall of the kingdom Plantae is made of cellulose. Thank you so much. So it is complex structure, means they are blanched, means you see they are blanched, their leaves, many organs are there, means they are multicellular. The mode of reproduction, usually they are sexual and asexual, and when you go to the level of their cell wall, when you go to the level of the cell of the plant, usually they have vacuole, which is large vacuole in the plant cells. I hope those are the characteristics of the kingdom Plantae. I hope it is clear because this one I have summarized very well. Take time and review. Take time and review. Take time and review. Take time and review. The key point here is to know the difference between one kingdom and another. So let we have the kingdom Animalia, which is fifth kingdom, and then after this we're done of the kingdoms, 5 kingdoms in this world. So you should understand, means there's different animals in this world, okay? But all of them, they are similar characteristics they are sharing. So number one, all of the animals, they are multicellular and they are eukaryotic, means they have many cells and also they have true nucleus, okay? Now all the animals, there is no animals which have chloroplast, means they do heterotrophic nutrition, means they don't make their own food, okay? They don't have cell wall. No animals in this world which have cell wall. So they have developed or not developed the nervous system. You know, the fungi, they don't have a nervous system. You know, like kingdom Monera, no nervous system, means nervous system also only they are in kingdom Animalia. So they have diverse forms, like they have wide variety of body plans and adaptation, means remember our hands, they can do many things. That the difference adaptation we did, and also means animals they are able to survive in many conditions. So that is what you call diverse forms. So the mode of reproduction mainly or primary are sexual reproduction, but there is some exception, some they do asexual reproduction. So it means that this kingdom Animalia, it is multicellular organism, they are eukaryotic organism, they do heterotrophic nutrition, they don't have cell wall, they don't have chloroplast. I hope you are understanding the difference between kingdom Animalia from other kingdoms. So I hope you are understanding these things very well. But before going there, let you have like I hope you are still understanding this figure here, okay? When we say kingdom Plantae, remember to say it is multicellular, and then remember to say it is eukaryote. So in plant, remember it is autotrophic nutrition, and then plant also they have chloroplast, and they have cell wall made of cellulose. Those are Plantae. I hope you are understanding key points there. Animalia, they are multicellular, they are eukaryote. So they don't have cell wall, they don't have chloroplast, but they have nervous system. They usually do sexual reproduction, but some exception they do asexual. I hope you are understanding also in Mammalia. In the kingdom of fungi, they don't have chloroplast. They have cell wall made of chitin. Mode of reproduction is a spore formation. Not only this one, they are usually heterotrophic nutrition, specifically saprophytic nutrition. I hope also you got this one. And now remember they are multicellular, they are eukaryote. So when you reach the level of the Protista, they are eukaryotic organism. They can be either unicellular or multicellular organism. They don't have chloroplast. Some they have cell wall, but other they don't have. Means some they are they do autotrophic nutrition or heterotrophic nutrition. I hope you are understanding. Now, when you come to the level of the bacteria, they are unicellular, they are prokaryotic cells. They have cell wall made of peptidoglycans. Bacteria also no chloroplast except some. Some they do their own food. Some bacteria like cyanobacteria they do autotrophic nutrition. But this bacteria they are unicellular, as I said. So I hope you are understanding these things. When you come to the archaea, remember that they can survive in extreme condition, sat or acidic condition, and then remember also archaea they don't have cell wall. I hope you are understanding the difference. So that's all about the kingdoms: Animalia, Protista, Fungi, Plantae, and so on. So after understanding the kingdoms, we have understand that the one kingdom there, they are called Monera. So when we say kingdom Monera, usually it's all about bacteria. So this bacteria here, they have ultra important, means bacteria, they cause disease. Many people, they know the bad effect of the bacteria, but some bacteria, they are very useful in many ways. So among the following ways, let me highlight some here. Bacteria are used in biotechnology. When we say biotechnology, I'm going to teach you biotechnology in senior 6 biology. We are going to see that some bacteria are used to make the like insulin in forms of what you call genetic engineering. So means this biotechnology, it's like production of ethanol, acetone. Organic means all about the industrial manufacture, making the different things by using the bacteria. So you can use bacteria also in forms of genetic engineering by doing what you call manipulating genes and the production of the human insulin. Means bacteria are very, very important in making the insulin, which will treat patients suffering from the diabetes. So bacteria are very, very important. So bacteria also are helping in breakdown of the organic matter. To make your organic matter be decomposed. Remember, bacteria is the one of the decomposers, means they have the ultra important role of breaking down the dead matter organisms. So I hope you are understanding these things very well. Let me have the marker also. When we come to the level of what we call nitrogen fixation, bacteria like Rhizobium is there in the legumes like soja beans roots, where it's going to make converting the nitrogen into nitrate so that this nitrate can be fixed in the soil and then improve the soil fertility. Means one of the importance of bacteria, it's improve soil fertility by fixing nitrogen in the soil, where it converts nitrogen to nitrate. So this Rhizobium is a kind of the bacteria. So some bacteria like Escherichia coli, E. coli, they synthesize vitamin B and vitamin K in small intestine of the human being. Means once you don't have the E. coli in your small intestine, you're going to have the problem of absorption of some vitamins. So bacteria and ruminants secrete cellulose for cellulose digestion. Yes, I hope it is clear. This is usually cellulose is not for human being. This is like for cow. For human being, this E. coli, it helps in absorption of the vitamin K, not B. So there is biological control, where like some bacteria, which is like a Bacillus thuringiensis, are used for the pest control agents, means environmental friendly, where you can introduce this bacteria now to make other organism being not affecting our plants. Pesticide. So there are some diseases of the bacteria in human being and also plant or animals. Usually the diseases caused of bacteria are important, but what is very, very important is just to list out which diseases are caused by bacteria, which diseases are caused by virus, things like that. So by having here a common bacterial disease, pathogenic bacteria, some bacteria are pathogenic, other are not causing disease, are like pneumoflora, means harmful bacteria causing diseases in human or animals or plants. Means bacteria can cause the disease either in human being, animals, and even plants. So here I have summarized for you the diseases caused by bacteria in human beings. So number one is called cholera, caused by what you call Vibrio cholera. This is the bacteria which causes this disease. Mode of contamination means the way in which this bacteria is entering in the form of the eating contaminated food or water or some vector. Signs and symptoms are here: vomiting, hydration, and all of this disease, cholera, dysentery, typhoid fever, all of those bacterial diseases are caused by poor hygiene. Means how can you prevent? It's just to improve your sanitation. Just all of these all of these diseases are how can you prevent? Just improve sanitation. How? Washing hands before eating, boiling water, proper waste disposal, means everything regarding to the hygiene. Just you are preventing those diseases. Cholera, dysentery, typhoid fever, and so on, all are the disease caused by the bacteria. Like cholera caused by Vibrio cholera, dysentery caused by Shigella, typhoid caused by Salmonella typhi. Those are mode of contamination are eating the food which is contaminated with one of these organisms. So their symptoms are going to be also similar, but like when your stool is watery, means look like water. Specifically, maybe you can think that you have a cholera. Like because you are roasting many water, dehydration means you're going to roast water, even vomiting. For dysentery, your stool is going to contain blood. Once you have your stool containing blood, you have to go to attend your hospital. Some abdominal cramps, and also this one, typhoid fever. There is high fever and the headache and then even fatigue. So not only human being but also bacteria can cause disease even in the plant. Here also there is the disease of the bacteria in the plant. One is common scab. This one, this is caused by this bacteria usually in potatoes. There is bacteria blight coffee. Usually this is the disease of the bacteria specifically in the leaves. There is another disease called the black lot of cotton. Also is a disease of the bacteria. Also it affects entire plant, means whole plant is affected. There is another disease called the blight of the beans. Usually it is a beans. They affect the beans specifically in part called the leaves. So this is the disease. At least if they ask you what are the two diseases caused by bacteria in a human being, you have to say cholera and even dysentery. And also if they ask you what are the two diseases caused by bacteria in the plant, you have to say like a common scab and also blight beans, and even what is also easier for you here. I hope you are understanding. So I hope everyone here, student, he wants to get a positive grade in biology. How to get a positive grade in biology? Just you have to stay consistent. You have to sit down without just going with what is going. It means once you know what you want, sit down and read things from 1 to the end. So that you can be sure that you can get a grade biology. So to get good grade, it is not accident. Work hard. Stay tuned. So I hope regarding to the classification in terms of domain, you have seen that there are 3 domains: Archaea, Eukarya, and then bacteria. The kingdom also we have seen 5: Plantae, Animalia, Protista, Monera, and fungi. I hope we are done. But also you can ask yourself, does virus have kingdom? Answer is no. There is no kingdom in the virus. Means virus don't have kingdoms. Means virus is not living things. But some crazy scientists, they say that virus, they are living things. Other, they don't say that this is not living things. Means there are some factors. So what is virus? Virus are a cellular microorganism. Means they are not cell. When you say a cellular, means it is not cell. It is not cell organism. It is visible only under electronic microscopy. So in the next unit, unit 3, I'm going to teach you what the unit 3 is called, the microscope. I will teach you what is electron microscope. But it is a materials which is used to magnify a small object. Means the virus, it is not a cell organism, you can only see under electronic microscope. In light microscope, you cannot see virus. So means virus is not classified as living organism. Why? Due to they don't have metabolism. And there is no cellular structure. There is no ribosome, no centriole, no nucleus, no everything. No mitochondria, no rough endoplasmic reticulum, nothing there in this virus. So let me continue by understanding deeply the characteristics or structure of the virus. So virus usually, let me draw for you. I have to come and then to draw usually bacteria, they have what you call capsid. Each bacteria, they have capsid made of protein. This one is capsid made of the protein. You can call it also even protein coat. And inside virus. Inside the virus, they have either DNA, they have either RNA. They don't have both. Means there is DNA virus and also RNA virus. There is no virus which can contain both DNA and RNA. Also, it is the difference between virus and the bacteria. Bacteria, they contain both, but virus, they have only either DNA or RNA. So, means there is here, which is capsid. Apart from the capsid, inside there is DNA or RNA or genetic material. And because of this genetic material, it's make this virus living organism. Only based on this fact, they have genetic material. They can classified as living organism. But based on that, they don't make any metabolism. Means there is no enzyme. There is no enzyme. There is no cellular organelles. They consider this one as an living organism. But based on that, they have nucleic acid. When we say nucleic acid, it is DNA or RNA. Because of this one, this virus, it is a living organism. I hope you are ready to talk about the structure of the virus. They have capsid. This capsid I have showed you. This one is the capsid of this virus. Inside there is the genetic material. Genetic material means some bacteria like influenza, so some virus like influenza, they have what you call envelope, but other they don't have envelope. I hope you are understanding the structure of this virus. Means not only this one, they have this tail with this one, these fibers. I hope you are seeing the structure of the virus. So characteristics of virus. Number one, you have seen that it is a cellular microorganism. Means individual virus is particles. Means it's like particles like umukungugu. It is particles. Much smaller than prokaryotic cell. Means they are very, very, very small. You have seen that you cannot even see it under light microscope. Only under electronic microscope you can see virus. You can see multiple choice question of this virus talking about the difference. Means is virus you can see under light microscope? Answer is no. Virus is only seen under electronic microscopy. Thank you. So means another characteristics, they lack cellular structure. There is no cell membrane, no cytoplasm. There is not any organelle, mitochondria, no organelles. Virus they have. That's why they are a non-living organism. So means, but you can ask yourself, virus is not organism. How does it cause disease? We understand that when it reaches human being, it make multiplication. How like stone can make multiplication in organism? Means they use what you call host. If they enter in your cells, they will replicate. They will make multiplication. Use your cells ribosomes. Another characteristics of the virus is parasite. It is called obligate parasite. Means they enter in living cells, which is host for survival. When the virus is outside environment, it is not living. Means there is no any change regarding to the they cannot multiply under outside, But when they are inside the living cells, they can multiply. So, genetic material, they contain either either DNA or RNA, and some they contain which can make them to evolve. You know, some virus, they make evolution. Which kind of evolution means because of their DNA here inside the capsid, we have seen that there is either DNA or RNA. This genetic material, you know, it is arranged in the forms of genetic code. Means once there is a mutation in this genetic material, your virus is no longer the previous one. Means it is already changed. So the classification of the virus are based on capsid. Either it is DNA or RNA. Means they can either have RNA virus and DNA virus. Means other we can classify based on the envelope. We have seen means if we say that this is capsid, this one there is genetic material. Some virus they have another cover called the envelope. Means there is some virus which have envelope and others don't have envelope. Means there is enveloped virus and an unenveloped virus. There is DNA virus and the RNA virus. So I hope you are understanding this classification of the virus. So after understanding this, we have seen that in the virus, we have seen the definition that it is not cellular microorganism. It is not visible under electronic microscopy. So we have seen that virus is a cellular microorganism. Means it is visible only under electronic microscopy. It is not living organism because they don't have enzyme, they don't have any cellular structure. I hope you are understanding why it is not living organism. So it is living organism for only one reason, because they have nucleic acid. Means because of this nucleic acid, when it is entering in a human being or any host or living cells, they can make multiplication as living organism. So in the structure we have seen two things: capsid made of the protein. Inside the capsid we have seen that there is genetic material which is either DNA or RNA. We have seen also some bacteria, some virus, they are enveloped. They have envelope. Other they don't have. Regarding to the characteristics, we have seen that the virus, they are very, very small. Virus, they don't have any cellular organelles. Virus is obligate parasite, and also virus, they can make evolution, means mutation. I hope you are understanding. So, by regarding to characteristic classification, means some of you have some virus, they are DNA virus, other RNA virus, depends on the genetic material which they contain inside the capsid. Other they are enveloped virus, other they are non-enveloped virus. I hope you are understanding this. If you are finding this video helpful, you have to make subscribe, comment and share, so that those students who are dealing with Candida Libre, they can come and watch this video and now get a positive grade in biology. Thank you. So some example of the virus, there is a COVID-19 virus, means coronavirus. I hope you are familiar with this coronavirus, which is the pandemic of 2020. When we say pandemic, it's all about the world distribution. It is not only COVID-19. There is Marburg virus. You can say there is adenovirus, Herpes. Means viruses are quite many, but either virus, they are RNA or DNA, or they are envelope or an envelope. So I hope you are understanding. So virus, they can cause the disease. As we have seen bacteria, they can cause the disease to human beings, specifically cholera, typhoid and so on. And also this virus, they can cause disease to human being. For example, there is HIV AIDS, influenza, which is Vichurane, chicken pox, common cold, COVID-19, can be the rabies, MERS, hepatitis. Hepatitis is caused by virus, hepatitis, a disease of liver, and also even polio. So here it is a structure of someone which is affected by one kind of the virus. Virus is not good to human being. So the mechanism in which the virus are causing disease, they can either come and automatically make what we call cell death. Also, they can come and disturb your homeostasis. When we say homeostasis, it is the maintaining internal environment in balance or in check without changing. Means once your temperature regulation, your glucose regulation, your things regulation is disturbed, means you're going to have disease. So means there is only just mechanism number one. It can be either which virus cause disease, either cell death or hemostasis disturbance. So some virus also, they come and then remain in your body forever, what you call latent. Means once you are immunocompromised, means once your immune system is your immune system capacity is declined, means that virus, they can re-emerging later. They can come back once your immune system is not strong. For example, here an example, there is what you call HIV. HIV is one example of the virus. If they entering you, they can keep there forever, like 8 years, without knowing that you are HIV positive. Once your immune system capability is decreased, now you're going to have the symptoms of HIV. So I'm requesting you, all of them, where you are, go and test your HIV to check if you don't have the HIV, which is not showing the signs and symptoms. So again, another example of the virus, there is human papillomavirus, which causes inkondo y'umura, hepatitis B virus, which are linked to the cancer. I hope you are understanding this. Virus is not good, they cause several diseases. You should memorize which diseases are caused by virus, at least 3, which diseases are caused by bacteria. I hope you are understanding. Very another important, very important, but which is last but not least, we should understand introduction to dichotomous key. This topic is complicated, too many students, but I'm going to make it like a piece of cake. When we say dichotomous key, what are we talking about? Dichotomous key, it is key. It is key as usual, okay? Dichotomous key is a biological identification tool. It is a tool which is biological identification, which used to classify organism. Remember, our unit is introduction to classification. How can you classify organism? To say that this organism is the kingdom Animalia, this one is the kingdom Plantae, kingdom Monera. What are we basing on? Let me answer you. We are basing on physical characteristics of the organism. One, they have the chlorophylls, other they don't have cell walls. Means based on those characteristics, we are classifying the organism. But the problem, what are the tools we are using? To say this is animals, based on maybe, when you say animals, Animalia, kingdom Animalia, we have seen should be multicellular, eukaryote, and so on and so forth and so forth. But we are not only ending on the kingdom, also kingdom is subdivided into phylum. Remember, among the popular phylums in the kingdom Animalia, it is Arthropoda. Not only Arthropoda, when we reach on the kingdom phylum Arthropoda, phylum also is subdivided into 2 classes. Phylum Arthropoda, they have 5 classes. They can be either crustacean, arachnida, Chiropoda, Dipropoda, or insecta. Means to classify all those organisms. Means to classify the phylum Arthropoda. What are you basing? Either physical characteristics, you can see. One, they have antenna, other they don't have antenna. Okay, those are physical characteristics. One, they have head, thorax, and abdomens. Other, they have what you call cephalothorax and the abdomen only. Means you can base on physical characteristics, and now based on those physical characteristics, you can classify. Means once you have antenna, 3 body part, 6 legs, you are insect. Very good. Once you have 2 body parts, you want the cockroach or the spider. Once you have 2 body part, 8 legs, I don't know where there is 8 legs, 8 legs, you are a arachnida. Once you are like you have like 8 or more legs, you are a crustaceans. Once you are myriapods or like Chiropoda or Diplopoda, means there is many ways, there is many physical characteristics we base on, so that we can classify the organisms. So the science, which the tools we are using to classify the organism, is called dichotomous key. I want you to take the definition of this dichotomous key. It is a biological identification tool used to classify organisms. Once you don't know what is dichotomous key, don't say you know anything in this unit. Yeah, I'm saying truth. So this dichotomous key is composed by a series of contrasting statements. Means they use a many series. Means based on the antenna, we can call this one. Based on the number of legs, we can call this. Means there is a many series of contrasting statements. Based on the body part, this one is this. Based on these things like this. So I hope you are understanding. So they are based on the contrasting the statement. So that guide identification based on the observable characteristics. I want to highlight these things. You should understand that this dichotomous key, they are based on the observable characteristics. Okay, means we are classifying our organism based on observable characteristics. Means what is dichotomous key? Dichotomous key is a biological identification tool used to classify organism based on what? Based on observable characteristics. I hope you are understanding. So if you are understanding this, give me a comment which says that I understand. So like let's continue by understanding the key characteristics. Means what are those key characteristics? I told you should see observable features, morphology, like shape, color, and structure. Now, what are the types of the characteristics? Is it qualitative, number? Sorry, when you say qualitative is not, number is quantitative. Is it a number? How many legs? How many antenna? How many wings? That things means those are the quantitative characteristics of the organisms. Means based on those you can classify any organism into their classes. Okay, thank you. So based on the qualitative, their legs, how they are appearing. When you say quality, how they are appearing. Are them having some things I can say like something which is not good, some things which can unique like a color or what? When you say qualitative, you can say like a color. You can means it is not you cannot means qualitative things. It cannot be put in number. Like legs. Legs are quantitative because you can say that 6 legs it is insect. 2 pair of antenna, means those things they are quantitative. But when you say so they are quantitative. When you say qualitative are things like their color. You can say that I have 6 colors. You cannot say I have this thing. No, means qualitative. They are like shape of abdomen, nature of the legs. They are nature. How are them appearing? Okay. It means based on this, you're going to make dichotomous key. So this is all about dichotomous key. And I know many students, they know definition, no problem. They know that they're based on physical, observable characteristics. Those physical characteristics, always they know. The problem they comes when they reach, they meet with the examples of any figure like this, and then ask them, construct dichotomous key. It become a problem. But as you have understood that it is composed by a series of contrasting statements, you should like take like this one. Come. Now, based on the antenna, this one means maybe you have this one they have antenna and this one they have antenna. Means based on the antenna, you cannot distinguish these two organisms. Okay. You should say this is the number one. This means based antenna or no antenna. Okay. Because based on antenna, many antennas are there. You should say the go to 2. Okay. Go to 2. Maybe on number 2, you are going to based on the number of the body part. Means having 3 body part. And then you see this one, they have 3 body part. No, maybe no other organism which have 3 body part. You say that if this maybe is A, you say means 3 body part, it is A. Okay. Now other more than 3 body part, if there is, you can say go to 3. Continue like that, having the series of contrasting statements until you're getting the unique features of each organism. But the ideas you should start by the large characteristics, huge characteristics. So let me continue by doing this example. Example here is asking to construct the dichotomask of this figure. You are having the rifts here. You are asked to construct the dichotomask. I hope you are understanding. So after finishing this example, also this video is going to be finished. And then I hope you should take a pen and understand how I'm doing. So like example here is to construct the dichotomask on this figure here. Okay. So it means how to do it. First things you should look at your figure or your image and use your observable morphology. Means the observable characteristics. What are the characteristics? If I'm seeing very well, those are the rifts. Okay, good. Those are the rifts. After understanding that this one are the rifts, you know in the rifts we have only two types of the rifts. Are compound rift or simple rift? Yes, maybe. Suppose maybe I have more than two simple rifts. You should check another things. Venation and so on, apex, margin. Let me have look how to construct this dichotomask. You should go with me. You should make dichotomask to identify. Means dichotomask, it is a biological tool which is to identify the organism based on observable characteristics. Means you are going to observe those organisms here and now make their classification. Thank you. So essential guidelines here use observable morphology characteristics. So either if venation, margin, when we say venation, is the arrangement of the veins along with their main veins. Margin, apex, and lamina, and petiole. So I will help you to understand all of this. First things to consider, you should consider number one. You should consider is the rifts. Means, as they have said that, it is a series. They told you that it is the series of the statement. Statement number one, type of the rift. Is the rifts compound or simple rifts? Okay? You should ask yourself, is the simple rift or the compound? When we say simple rift, it is usually rift with one main vein and one lamina, which is not separated. But when we say compound rift, we have many riflets, or means more than one riflets on your main veins. I hope you are understanding types of rift. Means based on this one, statement number one, you will come and then you will say number one. Statement number one, you will say that it is compound rift or simple rift. Sorry, this is compound simple rift or compound rift. Okay, statement number one. So I hope you are understanding. If you are seeing very well, this one compound rift. This one is compound rift, and this one is compound rift. Okay, and even this one is compound rift because means the rift are only one. There is only one lamina around the main vein. Only one lamina means both B, E, and G, they are a simple rift. Means they are rift, they have the one lamina, means they are simple rift. I hope you're understanding what is a simple rift. Now other remainings are compound rifts. Okay. Means like point number one, we can say that simple rift, you should go to 2. Okay, means the simple rift is not able to distinguish those three rifts. You should come here, number 2, what you call types of venation. Okay, types of venation. If you are remembering venation, there are two kind of venation. So how am I going to do it? So two kind of venation are there. Means either rift, there is 2 rifts here in front of me, okay, and this is main vein. Okay, there is another rift here. Now there is also main vein. When the veins are like this, are a network, are not parallel to the main vein, are not parallel, are called the reticulate venation. But when the veins are parallel to the main vein, This venation is called the parallel venation. Means after understanding, I have seen there are three reefs which are simple reefs. But among those simple reefs, some I can distinguish them based on types of venation. Either reticulate venation or parallel venation. So in terms of the venation, on number 2, okay, I can say in my BC, BEG, all of them are compound reefs, but G, there is one main vein and another parallel veins. Okay. Now, on this point number 2, types of venation, I have reef G, okay, which is parallel venation. I hope you are understanding, okay. You have seen that how I seen G means point number 1, compound reef, simple reef or compound reef. Simple reef, I'm not being able to distinguish compound reefs. I said go to 2, okay. Now on 2, I said either a venation, it is arrangement of the reefs vein in the reefs, okay. You know, there is either parallel venation or reticulate venation. Based on the venation, which is a parallel venation, I only remain with G, which is a parallel venation. Other remaining are not parallel venation. I hope you are understanding. Okay. So now after finishing this, okay, I can say I'm going just to continue, but G, I have already identified G, which is simple reef and parallel venation. Already there are no other reefs which have simple reef. Let me go in the other things which called digitate or non-digitate. So when we say digitate or non-digitate, digitate reefs are reefs with the multiple lobules, leaflets radiating in the single point, resembling the finger-like. If you are seeing this one, these reefs are looking like fingers. They are like finger-like. Okay, are called the digitate reefs. It means reef can be either digitate reef or non-digitate reef. Okay. Now when I reach here on the digitate reef, this one A is already identified. This one is identified. Means point number 3 is to talk about digitate or non-digitate. Means this one is digitate reefs. Okay. Which is reef A. Okay. So now based on the non-digitate, still now this E and B are still non-digitate. Their ropes are still okay. They are not divided. They have only single lamina. Okay, I hope you are understanding. So means I should say that A is digitate. Okay. Now on non-digitate, I cannot speak. Okay, I still have B and E, which is non-digitate. I should say you should go to E. No. So you should go to 4. Okay. In 4, we will talk about what we call reef margination. There is a smooth and rough. This is rough. And this is smooth. It is smooth. When we say smooth, there is nothing like this, okay? But when we say smooth is this one, which is smooth like a sima, kabakanyerera. While this one, kabakari ibintu bivunaguye, okay? When you look at the two reefs, E and B, when we say margination, which is rough, this E is identified. While this one, which is B, is smooth, and now is identified. Okay. I hope now those compound reefs we are done with them. Based on their reef margination, now you can separate E and B. To say that this E, it is simple reef, types of venation, reticulate venation, types of digitate, it is non-digitate, okay? In the types of margination, this E is rough, while this one is smooth. I hope you are understanding. So let's go to the riff, what we call rifflet. When we say rifflet are like this, means this is vein. Now the vein is subdivided into other riffs. Okay, it is one riff, but this riff is extra subdivided. Okay, is extra subdivided. Okay, those are the rifflet. Based on the number of rifflet, you see some they have more rifflet. See, many rifflets are here, but this one only 3 rifflets. Here also there are many. Based on the number of rifflets, you can say this one is 3 rifflets. They have 3 rifflets. We will say this one they have 3. Some they have 3 rifflets. Some they have 3 rifflets. Other they have more. Means this one is identified. Okay. Now based on bipinnate. Okay. When we say bipinnate, it is a central vein with a side branch. While it means there is a riff look like this. It is riff which have other riffs like this, like this, like this, like this, like this, like this. Yes. This is the riff. This riff is called pinnate. While there is another riff look like this. It is riff which is like this, and they have another branches. Okay. Another, and then also those branches also they have the riffs. Okay. Also they have the riffs, many riffs like this, like this. You see, it is this riff which have only one main vein and another extra riffs are called pinnate, while this one which have the main vein and also another main vein which is also have where to have riff is called the B. Means pinnate only one. Okay, but here they have 1 and 2. Okay, based on this one you can see here there is one main vein. Okay, and there is other things, and there is other riffs like this. But here you see it is like this only like this. Okay, means this one it is pinnate. Okay, while this one is bipinnate. So I hope I tried my best. And then you are understanding what I'm saying. So this is the answer, but I have a very summarized answer which is going to be flow. So this is an example we're just focusing about, and then here there is the answers which is very clear. I hope it is clear for you. Okay. So I hope you are seeing very well. Okay. First thing to consider, number 1. Okay. First thing to consider, it is simple or compound riffs. If it means here based on the simple, okay? Based on the simple, I told you can't distinguish any. You cannot distinguish because simple they are many. They are 1, 2, 3. Okay? You should say sir. Go to characteristics number 2. Okay. Only characteristics means they are going to be simple or compound. Let me take simple. Okay? Now they can go to characteristics number 2. Okay? Now on the characteristics number 2, you should say types of venation. If it is parallel venation, okay? If it is parallel venation, only one G is there. Okay. That's why on the parallel venation we already done to say that on point number 2 G. Okay, which is the okay. You see, it is parallel venation as I told you. Their veins are parallel as I told you. Okay. You should also say if it is network, means their network as I told you, their network venation. Okay. To say that this one, you see, this one and this are network venation. Okay, means you are not able to distinguish the two. Okay. You have to say go to characteristics number 3. Characteristics number 3, as I told you, it is simple digitate or non-digitate. Simple digitate, I told you that it is the riffs which is look like fingers on the hands. Okay. You are seeing here when you say the single digitate, now A is identified. That's why here you say that A. Okay. Now A, these characteristics, it is now identifying no other which is digitate, which has a finger like. Okay. Now, when we say like non-simple digit, okay, which don't have, this one don't have a finger like, and even this they don't have a finger like, okay. You have to say this one is not go to characteristics number 4. I'm still in the compound leaves. Characteristics number 4, you will see there is a smooth margin, okay. Remember, this one is digitate, non-digitate. What about their margin? Margin are smooth or are rough? This one is rough, this is smooth. This one which is smooth is B, now is already identified. That's why when we come here on the smooth, B already identified. No other which fit this condition to be simple riff, network venation, and also to be smooth margin. That is, now it is already done. Now on number 4, when we say scarlet margin or rough margin, it means also this one is identified. This one E is identified because no another which is compound riff, reticulate venation, and also non-digitate, and also which is rough. E is already now identified. E is also identified. So now we are on characteristics number 4. But all simple riff, we are done to classify them. Let me go to compound riffs. Okay. So in compound riffs, I have many, but by analyzing my compound riffs 1, 2, even 3. By analyzing my compound riffs, okay, I should analyze them based on the number of leaflets. Leaflets, I told you, are those things which come in here, okay? Leaflets, trifoliates which have 3 leaflets and more than 3, okay. Now they can be either leaflets which is 3 or more than 3. If it is 3 leaflets, I hope this F it have 3 leaflets. It is now already identified. Okay. It is now already identified. Okay. F. Now when you go to more than 3 leaflets, okay, we are remaining with this and this. You see, there is more leaflets. You can say more leaflets C. No, we still have D also more than. Means the condition of more than 3 leaflets is not able to distinguish the D and C. What to do next? You should have to go to another separation points. Okay, you should say go to 6 characteristics. Characteristics number 6 is P-net or B-net. If you remember very well, okay, I told you that P-net is like this, B-net is like this. Okay, if you look at those riffs very well, you see this one is the P-net, this one is the P-net. On the characteristics number 6, when we say P-net, this already the P-net is also identified. The P-net, which is C, is also identified. I hope you are understanding. And now that's all about the Quatermass key. Just go and highlight your characteristics to distinguish them. Okay. Starting from the general characteristics. Okay. Which is simply separating them in general. Like you say, simple riff and compound riff means each riffs here, they are either simple or compound. Thank you. Now, in that simple or compound, in simple, we have types of venation. Take venation. Some they are parallel, other are network. After finishing characteristics of the venation, take the digitate. Means if the riffs are separate or not. If this one not able to distinguish all of them, take the margin. Means your riffs, they have margin. How their margin are look like, are their quality. Is it smooth or is rough? If it is smooth, you see, and then you observe, then you conclude. Now, come again. Now, you should make more characteristics so that you can identify your organism. I hope I make this video fun. This is all about Unit 2, Introduction to Classification. I explain you the things. If you have seen that, it is very, very important. Make subscribe, comment, and share. But if you read very well, you are going to have a positive grade biology. Stay tuned for next video, and then encourage other students, which is specifically those who are studying Canada Libre and those who are in external who are in external school. Maybe any student who are studying MCB, PCB with the smartphone, just browse my YouTube channel, then enjoy. Thank you so much. I love you.